Welcome back. In the previous session, we studied the requirements of protein biosynthesis. Now, in the second session of today's class, let us study the first step of protein biosynthesis, that is initiation. Coming to specific learning objectives, you have already learned the definition of translation process, the requirements of protein biosynthesis, and the formation of charged tRNA molecules. Now, in this particular session, at the end of the session, you should be able to discuss the initiation of protein biosynthesis in eukaryotes with the help of a neat labeled diagram. Like transcription process, the protein biosynthesis can also be described in three steps, that is initiation, elongation, and termination. The information required for protein biosynthesis is present in the mRNA, and the mRNA has a 5' end and 3' end, and this information is decoded from 5' end to the 3' end. The first amino acid which gets added during the protein biosynthesis forms the N-terminal amino acid, and the last amino acid which gets added forms the C-terminal amino acid. Incidentally, the protein's amino acid sequence is also read from N-terminal to C-terminal end. However, post-translationally, the N-terminal amino acids and the C-terminal amino acids may be removed in the post-translational process. Now, I'll be describing the protein biosynthesis in the eukaryotes. The mechanism in the prokaryotes is also similar. Wherever there is a difference, I will just highlight that difference. Coming to the first step of protein biosynthesis, that is the initiation. Initiation occurs when the ribosome selects an appropriate mRNA and the mRNA binds to this ribosomes along with the tRNA molecule. The ribosomes should locate the initiator codon, that is the AUG, so as to set the correct reading frame. So when this happens, the initiation process is complete. Now, initiation also occurs in 4 steps. The first step is ribosomal dissociation, then the formation of 43S pre-initiation complex, the formation of 48S initiation complex, and the formation of 80S initiation complex. Now, this numbers 43S, 48S, and 80S refers to the ribosomes, and remember, the structure of ribosome is made up of 80S, which has got 2 subunits, the larger 60S and the smaller 40S. Now let us study in detail the different steps of this initiation process. Now coming to the first step, that is the ribosomal dissociation. Ribosomal dissociation here, it means the 80S ribosomes are split into the 40S smaller subunit and 60S larger subunit. This occurs with the help of the eukaryotic initiation factors 3 and eukaryotic initiation factors 1A. So these two factors are called as dissociation factors, and they bind to 40S and stabilize the structure. The same thing I have put across here in the diagrammatic form. That is, you can see here, that is the 80S ribosome is split to form the 40S and the 60S. And this 40S is stabilized here with the help of these eukaryotic initiation factors 3 and 1A. Whereas the 60S is stabilized by eukaryotic initiation factor 6. Even if you don't remember this eukaryotic initiation factor 6, it is all right. But you should remember the two dissociation factors here, that is the IF3 and IF1A. Now, coming to the second step of initiation, that is the formation of 43S pre-initiation complex. This occurs when a ternary complex consisting of the eukaryotic initiation factor 2 along with GTP and the initiator methionine tRNA binds to the 40S complex. Now, to begin with, first a binary complex is formed when the GTP binds to the eukaryotic initiation factor. So this two together forms the binary complex to which the initiator methionine tRNA binds. Now there are two tRNAs for methionine. One is the initiator methionine tRNA and another tRNA is for all the other methionine residues present in the protein structure. These two tRNAs are structurally different, but they charge the tRNA by the same enzyme amino acid tRNA synthetase. Now, the difference here between the prokaryotic and eukaryotic step here is in the first step of this binding of the methionine tRNA, in case of prokaryotes, instead of methionine tRNA, it is formyl methionine tRNA. That is, once the methionine tRNA has been formed in the prokaryotes, it undergoes addition of a formyl group from tetrahydrofolate with the help of a formyl transferase enzyme. Whereas in the eukaryotes, it is only methionine tRNA. Now the same thing I put across here again in the diagrammatic form. So you can see over here that there is a formation of a ternary complex. So the ternary complex here is made up of the eukaryotic initiation factor 2, which is attached to the methionine tRNA, and in turn this eukaryotic initiation factor 2 is having the GTP. So this binding of this GTP is important, which we will see the later gets hydrolyzed in the last step. So this is the formation of this ternary complex. Now this ternary complex binds to the 40S ribosome to form the 43S pre-initiation complex. This is a downloaded diagram where it has shown as 43S initiation, but actually it is correctly 43S pre-initiation complex. The next step in initiation process is the formation of 48S initiation complex. Now this occurs when an mRNA binds to the 43S pre-initiation complex. Now binding of this mRNA requires a protein called as cap binding protein. Now recall that the mRNA has got at its 5' end cap made up of 7-methyl guanosine triphosphate, and there is a poly A tail. Now this cap binding protein, which is nothing but the eukaryotic initiation factor 4F, which in turn is consisting of three initiation factors, that is 4E, 4G and 4A. In this 4E is important for binding of this cap binding protein to the cap of the mRNA molecule. Now, apart from this cap binding protein, another eukaryotic initiation factor 4B is also important because this particular factor reduces the secondary structure or melting of the structure at the 5' end of the mRNA. That is, this factor has got a ATP dependent helicase activity and it reduces this secondary structure present in the mRNA. Now recently it has also been found that it's not only the cap binding protein and the cap, but the poly A tail is also important in effective initiation process. That is, poly A tail along with the protein, poly A binding protein is also important for the initiation process. Now let us see the same thing in the diagrammatic form. So in the previous diagram, we had seen the formation of 43S pre-initiation complex when a ternary complex comes and binds to the 40S. Now in the next step, in the formation of 48S initiation complex over here, the cap of the mRNA is bound with the cap binding protein, that is the 4F. Now this 4F is in turn made up of 4E, 4A and 4G. Now the other important protein here is the eukaryotic initiation factor 4B, which is important for reducing the secondary structure in the mRNA molecule. And this whole process requires a hydrolysis of the ATP molecule, which is not shown in this diagram. So an ATP is required here for this process of binding of this mRNA to the ribosomes. So this forms the 48S initiation complex. Now with this, the mRNA is bound to the ribosomes, but the binding of the mRNA may not be at the correct position. That is, for appropriate binding of the mRNA to the ribosomal machinery, there should be approximation between the codon and the anticodon. That is, the initiator codon here is the AUG, and the corresponding anticodon arm of the methionine should correspond. So therefore, once the mRNA is bound to the ribosomes, now the complex scans for the appropriate initiator codon. So this may occur with the 5' most initiator codon, but the precise initiator codon is recognized with the help of a sequence called as Kozak sequence surrounding the initiator codon. This is the Kozak sequence surrounding the initiator codon. So this Kozak consensus sequence is important for identifying the correct initiator codon so that the mRNA can get correctly positioned on the translating machinery ribosomes so that a correct reading frame is established. A similar sequence is also present in the prokaryotes and it is called as Shine-Dalgarno sequence. So this sequence is called as the Shine-Dalgarno sequence. So in eukaryotes, it is the Kozak consensus sequence, and in the prokaryotes, it is the Shine-Dalgarno sequence. So from the previous step, that is the formation of 48S initiation complex, the attachment of tRNA with the methionine along with the ribosome is complete. Now the initiation process gets completed when there is association of this 60S ribosomal subunit with this complex to form the 80S initiation complex. So this occurs with the help of the eukaryotic initiation factor 5, which hydrolyzes the GTP present onto the eukaryotic initiation factor 2. So remember right from the beginning, the eukaryotic initiation factor 2 is carrying the GTP, and during this last step, the GTP gets hydrolyzed to form the GDP. And also there is release of all the other initiation factors. So 1A, 3, 4, 5, 6, all the initiation factor gets released to form finally the 80S initiation complex. Now all these initiation factors gets recycled. The eukaryotic initiation factor exchanges its GDP with the GTP with the help of a GTP-GDP exchange protein, that is the eukaryotic initiation factor 2B. So this particular protein helps in the again recycling of this binary complex, that is the eukaryotic initiation factor 2, so that it can take part in the next cycle of protein biosynthesis. To repeat once again, the 80S initiation complex is formed when 48 initiation complex binds to 60S. Now this occurs with the help of eukaryotic initiation factor 5, which hydrolyzes the GTP to GDP attached to the eukaryotic initiation factor 2. And then during this process, all the initiation factors are released. Now at this point, the 80S ribosomes are formed and it has got the 3 imaginary sites. Recall in the previous session, I had told that the ribosomes has got 3 imaginary sites, that is A site, P site and E site. So in the initiation process, the amino acyl tRNA, that is the methionine tRNA, is at P site. So this is a diagram showing only the P site and A site. The adjacent E site will be onto the left side of the P site, as we will see in the during the elongation process. So you can see that the at the end of the initiation process, the P site is occupied by the methionine tRNA. During the elongation process, all the other amino acyl tRNAs comes and binds to the A site, and once it binds to the A site, there may be formation of peptide bond, and the P site is occupied by peptidyl tRNA. We will study this in detail when we go for elongation process. Coming to the last part of today's session, that is the regulation of initiation in the protein biosynthesis. Protein biosynthesis is a high energy requiring process requiring a large number of ATPs. So hence, whenever there is decrease in the ATP levels, that is whenever the cell is under stress, the protein biosynthesis is inhibited at the initiation step itself. There are two important regulatory proteins involved in the regulation of the initiation. The first one is regulation by the eukaryotic initiation factor 2. Now this protein IF2 consists of three subunits, that is the alpha, beta and gamma subunits. Now the alpha subunit can undergo phosphorylation with the help of various kinases. Now during the stressful conditions in the cell like amino acid deprivation, glucose deprivation, viral infections, heat shock, etc., the kinases gets activated, which in turn phosphorylates the 2-alpha subunit of this initiation factor 2. Now this phosphorylated eukaryotic initiation factor 2 binds more tightly with the GTP-GTP recycling protein, that is the eukaryotic initiation factor 2B. Now recall that this particular initiation factor 2B is important in recycling of the GTP with the GTP, that is the exchange of GTP with the GTP, so that it can regenerate back the binary complex of eukaryotic initiation factor 2 GTP complex. So once there is no regeneration of this binary complex, the next cycle of protein biosynthesis gets affected and hence protein biosynthesis is inhibited. So overall, the protein biosynthesis is arrested during the stressful condition, that is whenever there is a decrease in the ATP levels, which can occur due to a number of reasons as I already explained. Coming to the second important regulatory molecule, that is the regulation by eukaryotic initiation factor 4E. Now recall that this particular factor is a part of the cap binding protein 4F. Now 4E is responsible for the recognition of the mRNA cap structure. That is, this particular molecule is the one which is mainly responsible for binding of the cap protein to the cap of the mRNA molecule. Now, phosphorylated 4E binds more avidly with the cap of the mRNA molecule. So, therefore, anything which causes phosphorylation will increase the initiation process. And insulin, the hormone, and various mitogenic factors phosphorylates this particular initiation factor, that is the 4E factor. So, hence, that is the reason, one of the mechanisms by which insulin increases the translational activity in the cell. So overall, if I have to put in one sentence, so regulation of eukaryotic initiation factor 4E controls the initiation step. So with this, we have come to the end of today's class. So from the two sessions, we studied that protein biosynthesis occurs on the ribosomes attached to the endoplasmic reticulum are freely present in the cytosol. Protein biosynthesis requires a number of molecules, including the 20 standard amino acids, various RNA molecules, and various protein factors. So protein biosynthesis takes place in three steps, that is the initiation, elongation, and termination. So before initiation, the amino acids are activated by getting attached to the tRNA molecules. Initiation occurs in 4 steps, that is the ribosomal dissociation, formation of 43S pre-initiation complex, formation of 48S initiation complex, and formation of 80S initiation complex. At the end of the initiation, the mRNA is correctly positioned on the ribosomes and methionine tRNA is at the P site, and ribosomes are ready to begin with the elongation process. So we will be studying the elongation and termination steps of protein biosynthesis in the next class along with post-translational modifications. Thank you. If you have any doubts, you can always contact me.